Today what I'm going to talk about is the notion that to promote optimal brain health throughout the life course and perhaps reduce risk for age-related neurodegenerative disorders such as Alzheimer's and Parkinson's diseases, that it's important to challenge yourself intermittently with bioenergetic stresses. So exercise, when you exercise, that's definitely a stress not only in your muscles but your cardiovascular system. We think it also will affect adaptive stress response signaling pathways in your brain. In fact, it was shown a long time ago that exercise is a potent stimulator of BDNF production, which is a neurotrophic factor that promotes plasticity but also protects neurons against oxidative stress, metabolic stress, and so on. I'm going to talk a little bit about exercise. I'm also going to talk about fasting, which is another challenge to the brain and body. From an evolutionary perspective, if you haven't been able to obtain food, your brain better be working well, or you're not going to figure out how to effectively compete to obtain that food. And I'm going to talk about some of our work on intermittent fasting and its effects on the brain in models that are relevant to age-related neurodegenerative disorders. And then finally, I'm going to also talk about some recent work and perhaps more so a hypothesis of why it is that fruits and vegetables may be good for our health, including brain health. So during brain aging, there are a number of changes that occur in nerve cells that occur in other cells throughout the body, and these are pretty well established. For example, there's pretty good evidence during normal aging cells have problems generating enough ATP, so there's some problems in mitochondrial function. We've done a lot of work in my lab on neuronal calcium homeostasis. As you know, calcium is an important second messenger in neurons. It's critical for synaptic plasticity, learning, and memory. However, if neurons are not able to maintain calcium levels, and particularly restore calcium levels to low levels after action potentials or activation of synapses by glutamate, that can cause problems. Oxidative stress. There's a lot of excitement and activity in understanding how cells and neurons remove damaged molecules and organelles, and there's accumulating evidence that, at least to some extent, the impaired autophagy or perhaps other mechanisms whereby cells remove aggregated proteins may play a role in neurodegenerative disorders, but also in normal aging. In fact, if we look at some of these proteins that accumulate in Alzheimer's disease, A-beta and tau, or ALS, TDP-43, Parkinson's alpha-synuclein, we find that oxidative stress, impaired calcium handling, and impaired bioenergetics can actually exacerbate accumulation of these proteins. Okay, then also during normal aging, there's evidence that the ability of brain cells to respond adaptively to stress is impaired. For example, reduced neurotrophic factor signaling, but perhaps other mechanisms. We've done some work recently on DNA repair that suggests that during normal aging, there's declines in base excision repair in neurons, which may increase their vulnerability to accumulation of amyloid. So these different changes that are occurring during aging in combination with accumulation of these pathogenic proteins can lead to synaptic dysfunction and neural degeneration. Okay, that's the bad news. The good news is that there's emerging evidence, mostly from animal studies, but also from human epidemiological studies and even some clinical intervention trials, that exercise, energy restriction, doing what we're doing now, hopefully, if you're paying attention and thinking about what I'm talking about, you're imposing a mild stress on your neurons because they're active, and we think that's good to have intermittently use your brain cells, but it's also important to sleep. The last slide of my talk is going to be I'm going to go through, my main part of my talk is what are these adaptive stress response pathways that are activated by exercise, fasting, and certain phytochemicals. But then the last slide of my talk, I'm going to point out that we need a recovery period from these challenges in order to promote plasticity and optimal brain function. Okay, what else do I want to say here? Okay, some of the work we've done in my lab suggests that dietary energy restriction, particularly intermittent fasting, and also exercise can not only upregulate neurotrophic factors, but increase the production and activity of certain protein deacetylases. I'm going to talk about some recent work we've done on a mitochondrial deacetylase called SIRT3 with Will Bohr, B-O-H-R, who is Niels Bohr's grandson. That's just an aside. But he's an expert on DNA damage and repair, and his lab is one floor above mine. Together with him, we've found that exercise can upregulate certain DNA repair enzymes. We've also done some work, as have others recently, on mitochondrial biogenesis, which is, as its name implies, the process whereby mitochondria grow and divide, which is very important in neurons during development, but we think also in plasticity. Okay. I'm going to start out talking about the hippocampus. As most of you know, this is an important region of the brain for learning and memory, and the simple explanation for that is it's where our sensory inputs converge. So, example, if you're looking at my slide and listening to me now, simultaneously you're seeing an image on the screen and you're hearing what I'm saying, and that information is funneling into the hippocampus. So that information converges on synapses, and when that happens in a temporally coincident manner, those synapses are strengthened so that next time you may see a picture of the hippocampus and you may have an image of my face in your brain. So that's kind of a simple explanation, but that's true. And also this circuitry is affected in Alzheimer's disease. These inputs, these axons coming in from the entorhinal cortex, those entorhinal cortex neurons degenerate. Interestingly, the dentate gyrus neurons don't tend to degenerate in Alzheimer's, which is still a very interesting problem from the standpoint of selective neural vulnerability. However, the pyramidal neurons in the hippocampus do degenerate in Alzheimer's disease. Okay, so exercise and some work we've done on energy restriction, others have done work with enriched environments, have shown, provided good evidence that these pathways are more active during exercise, fasting, and certainly when you're intellectually engaged. And that increased activity increases BDNF production, which promotes the increase in the physical size and even the numbers of synapses in the dentate gyrus and also other regions of the hippocampus. As you know from work done in many labs, some people here, including Jason, working on this important problem, there are stem cells in the hippocampus that undergo self-renewal, and then they can stop dividing and differentiate into neurons, some of which become integrated into the dentate gyrus and receive inputs and may actually play important roles in certain types of learning and memory, particularly spatial pattern separation, which I'll briefly talk about in the next slide after this. So Henriette von Praag is a tenure track investigator in my laboratory, so she's a senior person. When she was working with Rusty Gage at the Salk Institute, she found that if you put running wheels in the cages of rats or mice and then look at neurogenesis by labeling proliferating cells with bromodeoxyuridine and then aminostaining, so the black dots are cells that have undergone DNA synthesis and divided. Bottom line is runners, there's increased proliferation of these stem cells, and then there's also an increased number of newly generated neurons that survive and become integrated into the circuitry. So these can be labeled with retrovirus. As an aside, this is my daughter and son stimulating neurogenesis in their hippocampus. And you can see on their face here that this is a stress. But I coached high school cross country for 7 years, and I'm also a runner, and I understand. During the race, you're saying, why am I doing this? But then within a minute or two after finishing their race, you feel great. You're ready to do it again. Okay, so Henriette von Praag, she sent a postbac fellow, Dave Creer. NIH has a program for kids after they get their bachelor's degree, they can spend a year or two in the lab. And so Henriette sent Dave to Cambridge in England to Tim Bussey's lab, and Tim was developing touchscreen systems so that he can test various aspects of learning and memory. And one thing that you can test is what's called spatial pattern separation, and I'll briefly describe how this works. So first of all, you train the mouse that if it touches its nose on the computer screen, and these are small boxes like this big, that'll get a food reward. So this is all food reward based. You fast the animals overnight, and so they're hungry. And then it takes a week or two so they learn that if they touch the screen, they get a food reward. Then you can teach them, if you put one lit icon on the screen, if they touch that, they get a food reward. That takes a couple more weeks. It's all automated, though. You just put the mouse in and leave it in there for 7 hours, and during that time, it's learning this. And then what you do is you put up 2 icons on the screen separated by a big distance, and the mouse, you can train it to touch. If it touches this icon, it gets a reward. If it touches this one, it doesn't. Then you decrease the distance between the icons, and as you do that, you decrease the distance, the animals make more errors. However, if you compare animals that have had running wheels in their cages for a couple weeks to those who are sedentary, the runners make fewer errors compared to the sedentary animals, and there was a correlation between that and BRDU labeled cells. Okay, then in my group, a former graduate student, Alexis Stranahan, did a couple studies to look at plasticity in the hippocampus with a focus on the dentate gyrus. The dentate gyrus is critical for that spatial pattern separation performance of that task that I showed in the previous slide. So in this study, what Alexis did, she'd previously shown in this Nature Neuroscience paper that diabetic mice, these are so-called DB-DB mice, they have a mutation in the leptin receptor. Leptin's a hormone. When you eat a meal, it's released from fat cells into the blood. It goes to your hypothalamus, and it tells you to stop eating, don't overeat. So these mice don't get the stop eating signal, and they overeat and become obese and diabetic. And they have impaired learning and memory in water maze. They have impaired LTP at CA1. No, Alexis did dentate gyrus synapse, perforant path dentate gyrus, and they also have reduced dendritic spine density. So in this study, what Alexis did, she had wild-type mice, diabetic mice, and she put them under 4 different environmental conditions. The normal laboratory housing conditions, which is couch potato, ad libitum fed and sedentary. Then she had a group ad libitum fed with running wheels in their cages. Daily calorie restriction, 30% reduction in calories, sedentary, and then calorie restriction plus running. Let them go for 3 months and then killed them. One hippocampus, she did Golgi staining to quantify dendritic spine density. The other hippocampus, she did BDNF ELISA to measure BDNF levels. And the bottom line is that both caloric restriction and running over this 3-month period, there was an increase in spine density in wild-type mice, and the combination of running and calorie restriction, there was a further increase. The diabetic animals had low dendritic spine density, which she'd shown before, and however, both caloric restriction and running increased spine density significantly in the diabetic animals. And there's nice correlations between the effects of these environmental conditions on dendritic spine density and BDNF levels. So what might be the relevance of this to humans? There are some humans who have BDNF haploinsufficiency. That is to say, they only have one copy of the BDNF gene, so they're like BDNF heterozygous knockout mice. And other labs had shown that BDNF heterozygous knockout mice, particularly as they get older, have impaired performance in learning and memory tasks. These study was done in teenagers that are either wild type for BDNF or heterozygous for the BDNF gene. And they tested their IQ in a certain test, and there was the kids with presumably with reduced BDNF levels have poorer performance than the wild type kids, if you will. And then, since this paper came out pretty recently and autism is a hot topic, they also tested these kids in using some autism spectrum disorder criteria and found that these kids with low BDNF levels have more autism-like behaviors than do normal kids. Okay, I'm quickly going to show you 3 slides. And this was this kind of a fun thing that I did based on Henriette's work and others' work on spatial pattern separation, because Henriette would show me her data on the simple spatial pattern separation task in animals, and I would ask her, you know, isn't everything the brain does, that, particularly human brain, that makes us uniquely human based on pattern processing, our ability to process patterns, whether visual, auditory, better than other lower animals. Okay. So whereas the hippocampus, which is important for these, what I would say are kind of simple, at least among mammals, processing patterns, what about the brain regions that have expanded in humans? So prefrontal cortex, which is involved in decision making, for example, and executive function. And then also humans have expanded cortical regions that are associated with visual inputs and processing auditory information, obviously the language areas. So these are some of the things that, as far as we know anyway, in some cases for sure we know do better than lower species and maybe correlated with these expanded brain regions. So I wrote this article on superior pattern processing is the essence of the evolved human brain. And it's just pointing out that everything our brain does is processing patterns, whether it's creating new patterns, imagining things that can exist, invention, and so on. And in fact, I organized a workshop coming up. There's this guy, Lawrence Krauss, who's a cosmologist, who started this thing called the Origins Project at Arizona State University, and we're going to do one on origins of pattern processing, which is, we'll have neuroscientists and then artificial intelligence, computer science, and robotics people discussing this kind of black box of our neuroscience knowledge of how does the brain process patterns once they're encoded. Okay, now let's get to some molecular and cellular biology. Okay, so there's been a lot of work obviously done on how exercise affects muscle cells, cardiovascular cells, and it's been shown in muscle cells that exercise will increase the number of mitochondria in the cells, and that makes sense. Then the cells can generate more ATP. So endurance athletes will have more mitochondria in the muscle cells that are involved in endurance behavior. And the way this is thought to work, and this is pretty strong evidence, that the exercise it activates a kinase called AMP, activated protein kinase, also increases free radical production, reactive oxygen species. So in your muscle cells, when you're exercising, the cells are generating a lot of free radicals, and there's a big increase in energy demand. And there's this transcriptional regulator, PGC1 alpha, that is upregulated by this kinase and by reactive oxygen species. And it will induce the expression of multiple genes that encode proteins that are critical for mitochondrial biogenesis. And there's at least a dozen such proteins that have been identified. And so we start out by asking a simple question. If we manipulate PGC1 alpha levels in neurons, does it affect mitochondrial biogenesis in the neurons? And two, if it does PGC1 alpha play an important role in, for example, responses of neurons to exercise and signals that are upregulated by exercise, such as BDNF? So I'm going to quickly go through some data, a paper we published several years ago in Nature Communications. This is work's done by Eiwu Cheng. What she did is she made viral vectors to either knock down or overexpress PGC1 alpha, and first in cultured hippocampal neurons, and then in vivo in dentate granule neurons. And I'll go through some of those data, and then I'm going to spend a little more time on this recent paper. Actually, just came out it was online in 2015, but it just came out in this month's Cell Metabolism, where we did some work in SIR2 knockout mice that hopefully you'll find interesting. Okay, so if we take dissociate cells from embryonic rat hippocampus, put them in culture, they start out as spheres, they adhere to the substrate, they grow neurites, and one of them differentiates into an axon, the others into dendrites, and this is a neuron 3 days in culture, this is one 10 days in culture, and the red here is mitochondria labeled with a fluorescent probe called MitoTracker red. The only point of this image is that as these neurons grow, there's more and more mitochondria that fill the growing dendrites and also the axon, so that number of mitochondria increase over time in culture. ATP levels increase over time. And this just shows we didn't find any change over time and development in culture in the size, the average size of individual mitochondria, or the size distribution. This slide just shows that we can reduce or increase levels of expression of PGC1 alpha. This is mRNA levels, this is protein levels. So we're using adenovirus. It also has GFP in it, so that any infected neuron will be green. And so this is just kind of confirmation at the immunohistochemical level that these two neurons here, this one and this one, which are green, we fix the cells then in immunostain sorry, where am I? Getting lost. And these have very low levels of PGC1 alpha immunofluorescence compared to these other two neurons that weren't infected with the virus. And so Eiwu infected the cells at about 3 days in culture and let them go for 10 days. And then during that time, synapses will form and dendritic spines will form. And bottom line is she found that compared to control infected neurons infected with a scrambled sequence, those in which she knocked down PGC1 alpha with siRNA, when she knocked down PGC1 alpha, there's lower dendritic spine density. If she overexpressed PGC1 alpha, it increased the spine density, and that's quantified in the paper, which I'm not showing you all those data here. And then she injected the virus into the dentate gyrus of adult mice, in this case to knock down expression of PGC1 alpha. And this is just showing, this is the cell body layer here, and immunostained with PGC1 alpha. The ones she knocked down, there's less fluorescence. So these are the cells that are infected in green. And then she let them go, again for 2 weeks in this case, and quantified some parameters of dendritic architecture. The dendrite length and branches numbers weren't affected, but the dendritic spine density was significantly reduced. So our interpretation of these data, both in culture and in vivo, is that PGC1 alpha and possibly mitochondrial biogenesis are critical for the formation and maintenance of synapses in the hippocampus. Then we went on to, since it's known that BDNF can promote synaptogenesis, we asked the question whether BDNF might affect PGC1 alpha promoter activity. And I'm not going to go over the details of the molecular biology, but we found BDNF increases PGC1 alpha promoter activity in this luciferase expression system. And that requires a Cre binding site. Others had already shown that was critical for PGC1 alpha promoter activity. Okay, then what we did is we found, as had others, that if we treat these cultured embryonic neurons with BDNF over a period of a week or so, we can increase the number of synapses, at least in this case determined by PSD95 positive puncta. However, if we knock down PGC1 alpha using the viral vectors, BDNF is no longer able to increase the spine densities. So this indicates BDNF may be critical for, PGC1 alpha may be critical for BDNF-induced synapse formation. It also suggests the possibility that environmental factors that upregulate BDNF could possibly increase the number of mitochondrial neurons in the brain. And we don't have the data to show that in vivo yet. Okay, now I'm going to talk about SIRT3. So these SIRT2s are protein deacetylases. The one that's been mostly studied is SIRT1, which is located mainly in the nucleus. There's another one in the same family, and it's an NAD-dependent enzyme called SIRT3, which is localized just in mitochondria. So in collaboration with David Geis at the NCI, he had made SIRT3 knockout mice, which interestingly don't have any obvious phenotype, at least in young animals. And we just took some brain tissue from cortical tissue from wild type SIRT3 knockouts, separated the proteins on a gel, and then did a Western blot, probing the blot with an anti-acetylated protein antibody. So the bottom line is there's hyperacetylation of many proteins in brain tissue of the SIRT3 knockout mice compared to wild type mice. And then we isolated mitochondrial and cytosolic fractions and found that there's hyperacetylation of several mitochondrial proteins, but not any detectable hyperacetylation of cytosolic proteins. We found that by doing immunoprecipitation using antibodies to SOD2, a mitochondrial antioxidant enzyme, and cyclophilin D, which is a protein involved in formation of the permeability transition pores, which are involved in apoptosis. These seem to be two substrates of SIRT3 in the mitochondria, because when we IP SOD2 and then probe with the acetylysine antibody, we see hyperacetylation in tissue from SIRT3 knockout mice, and similar with cyclophilin D. And in fact, the reason we focused on these two proteins is that other labs who, not neuroscientists, and in other cell types had shown, provided good evidence that these two proteins are in fact direct substrates of SIRT3. Okay, then in the cultured neurons, hippocampal neurons, we ask a simple question. We expose the cells to increasing concentrations of glutamate. So low concentrations of glutamate will cause a transient calcium influx, similar to what's probably occurring at synapses. However, if you expose neurons to high levels of glutamate, you get calcium influx, but then the calcium levels are unable to recover. And actually, the cells can die by a process called excitotoxicity. So what we found is that we saw a biphasic curve in levels of SIRT3. So we exposed for 24 hours, we had glutamate, and then measure, actually it may be less. I think it's less. I think it's 6 hours. And then measure SIRT3 protein levels. So we see this biphasic curve. Low levels of glutamate increase SIRT3 levels, high levels decrease. Saw a similar thing with NMDA and canic acid, which activate the NMDA and kainate subtypes of glutamate receptors. Okay, so glutamate at low levels can upregulate SIRT3. We also found that in vivo, if we put running wheels in the cages of mice for 2 weeks, in this case we see significant increases in SIRT3 protein levels in cortex and hippocampus. Okay. Then going back to culture, we looked at vulnerability of SIRT3 knockout neurons in red and wild-type neurons to increasing concentrations of glutamate receptor agonists. And we found that the SIRT3 knockout neurons are more vulnerable to being killed by glutamate, kainate, and NMDA compared to wild-type neurons. Then we made an AAV with a SIRT3 cDNA in it, so we can overexpress SIRT3, shown here by Western blot, and this is in primary neurons in culture. And we find if we overexpress SIRT3, which is the black bars here, we can reduce the glutamate, we can essentially reduce excitotoxic neuronal death significantly in both wild-type neurons and SIRT3 knockout neurons. So we can rescue the cytotoxicity vulnerability phenotype by overexpressing SIRT3. Then we looked at excitotoxicity in vivo in the canic acid seizure model, and so we can quantify viable neurons remaining several days after canic acid treatment. And so down here in these graphs, it's quantifying surviving neurons in region CA1 and CA3 in wild-type mice is black, and SIRT3 knockout is the lighter color. So these are the animals given kainate here. These are animals injected with saline. So we see a lot of cell loss in both the wild-type and the SIRT3 knockout mice, but there's more cell loss, statistically significantly more in the SIRT3 knockout mice. Then we sent this manuscript to Cell Metabolism, and the reviewers were very interested in the exercise result, and we hadn't really elaborated on what's the mechanism whereby exercise is upregulating SIRT3. So what we did, since we found that glutamate will upregulate SIRT3 in culture, we repeated the running experiments, and we had separate groups of animals that were given a drug called MK801, which blocks NMDA receptors. And the bottom line, we found that whereas in the absence of MK801, running upregulates SIRT3 expression, when we block NMDA receptors, running is no longer able to increase SIRT3 expression, indicating that ongoing activity in the neuronal circuits in the hippocampus, we think, is mediating the effects of exercise in upregulating SIRT3. And then finally in this study, we also used a model that's relevant to Huntington's disease, where we administer a toxin called 3-NP, 3-nitropropionic acid, that damages striatal medium spiny neurons. So these green here, each green dot is a neuron immunostained with antibody to nu-N. So this is in wild-type mice injected with PBS, that is no toxin. This is wild-type mice injected with the toxin. This is SIRT3 knockout mice injected with the toxin. So you see there's less green here, meaning more striatal neurons have died, and that's quantified here. So there's far fewer striatal neurons survive exposure to this mitochondrial toxin in the SIRT3 knockout mice compared to the wild-type mice. So this is our working model here. There's still many dots that haven't been connected, but this is the working model that exercise, also intermittent fasting and cognitive stimulation, upregulate activity in neural circuits, increase BDNF expression, which actually in part could be through increased activity, excitatory activity, which is known to upregulate BDNF expression. And then it's been known previously that CREB upregulates PGC1 alpha, and we think CREB is mediating BDNF and also glutamate receptor activation, induced upregulation of PGC1 alpha. SIRT3 may be downstream of PGC1 alpha. It's one of the mitochondrial proteins that's upregulated by PGC1 alpha. And then in the mitochondria, so we get increased number of mitochondria through this pathway, through mitochondrial biogenesis, and here's a few of the proteins involved in that. And also, we think we get increased resistance of the mitochondria to stress through SIRT3-mediated deacetylation of SOD2, which increases the activity of this enzyme, and that's in the published literature. And then deacetylation of cyclophilin D decreases its ability to associate with and promote formation of the permeability transition pore, which may protect against apoptosis. Okay, so at the beginning I mentioned that evolution favors individuals whose brains are functioning well when they're hungry. I gave a TED talk on this, so if you're interested you can look at the TED talk, but I'm going to quickly show you a couple slides from that. There's a historical perspective on why fasting, or not why, but maybe if fasting might be good for cognitive function. So in the literature from a long time ago, not peer-reviewed, but there are famous, very smart people who claim in their writing that they thought much better and did much better work when they were fasting. Okay, and there's a lot of important quotes here that I think are very true. Our current health care system and biomedical training of physicians de-emphasizes what we know works and emphasizes what works less well. Patient comes in with insulin resistance, doctor says they're overweight, doctor says you need to exercise, cut back on your food intake, they come back the next year, they weigh more, now they're diabetic, the doctor says, okay, we're going to give you this drug, and they don't pursue doing what we know works, which is reducing energy intake and exercise, which can prevent and cure diabetes, there's no question, and we think it may also be good for the brain. Okay, so fasting. We published many studies beginning in the late 1990s where we take rats or mice, we divide them into two groups. One group fed ad libitum, the other group, every other day they go 24 hours without food, alternate day fasting. If one begins this alternate day fasting at this post-weaning age, the animals will live about 30% longer on average, so it extends their lifespan. And that was actually shown many years ago by other labs. What we found is in some of these toxin-based models of neurodegenerative disorders that I mentioned, and also more recently in some of the genetic mutation-based models, that intermittent fasting can be beneficial. So for example, we found that intermittent fasting protects against canic acid seizure-induced damage and 3-NP-induced damage to the striatal neurons in a model that's relevant to Huntington's disease. Actually then, when Huntington mutant mice became available, we took the Huntington mutant mice and found that if we start them on alternate day fasting early on, it slows the onset and progression of the clinical phenotype and the accumulation of the mutant Huntington aggregates in the neurons in the striatum and cortex. We did a study in what was called the 3X-TGAD mouse model, which was developed by Frank LaFerla, and we helped a little bit because we gave him the PS presenilin-1 mutant knock-in mice that he used to make these 3X-TGAD mice. But if we start these mice on alternate day fasting diet or daily calorie restriction when they're young, it greatly delays the onset of the cognitive dysfunction. We've done a study in stroke models that if, and in these cases with the excitotoxin, neurotoxin in the stroke model, we have to have the animals on the intermittent fasting before they're given the toxin or before we occlude the artery in their brain in the stroke model to see a beneficial effect. So this is kind of it's called hormesis or preconditioning. The idea is, and we've shown that the intermittent fasting can upregulate adaptive stress response pathways so that when the brain is subjected to more severe stress, whether it's a pathogenic protein like A-beta or synuclein or oxidative or metabolic stress, they're more able to resist the stress. Okay, and then we extended our animal work in several collaborative studies to human studies of intermittent fasting or intermittent energy restriction. And the first study we published was with Jim Johnson, who works with asthma patients, and his group took the asthma patients. They were all overweight, and put them on a diet, and it's a very rigorous diet that I don't think most people could follow. So they had them every other day, they only ate 500 calories. And they did that for 2 months. There were 12 subjects, only 2 dropped out, and the 2 that dropped out in the first 2 weeks. The other made it through the 2 months. But anyway, so they lost weight, their asthma symptoms improved, their airway resistance improved, and in my lab we measured markers of oxidative stress and inflammation in their blood, which over a period of a few weeks, and then by 1 month, big reductions in markers of oxidative stress and inflammation. Then we did collaboration with Michelle Harvey in women at risk for breast cancer. And in that study, she put the women on either what's now called the 5-2 diet, which is 2 days a week they only eat 500 calories, the other 5 days eat normally. Or we had a group that had a daily reduction in calorie intake of 25%, and we let them go for 6 months on these diets. Both groups of women lost weight, but the women on this 5-2 diet lost more belly fat, and they had a greater increase in insulin sensitivity. So there seemed to be some additional benefit, and the reason is in part because with fasting, or even short fasts of 16 hours, what will happen is about 10 to 12 hours after not eating anything, you deplete the glycogen stores in the liver, then mobilize fatty acids from fat cells, which then go into the liver and are converted to what are called ketone bodies. It's been known for a long time that ketones are good for the brain. In fact, ketogenic diets, which is a way to increase levels of these ketones by eating fats, but you can also do it by fasting, and actually fasting is better. You get higher levels of beta-hydroxybutyrate than with ketogenic diet. Anyway, so some of these collaborative studies led to a number of popular books. One is the alternate day diet, which came from the asthma study we did. The fast diet and the 2-day diet came from the 5-2 diet studies we did with Michelle Harvey. Actually, this is Michelle Harvey's book. I didn't write any books because they did a good job writing them, and I can't get royalties because I'm a U.S. government employee, so there's no monetary incentive. But help, maybe it'll help people's health that we had some input on in the early end of this. Okay, the ketones. So ketones, the main one that's generated when you fast is beta-hydroxybutyrate from the fatty acids that come from the fat cells. And then it gets into your brain cells, and it's a source of acetyl-CoA for the Krebs cycle, TCA cycle, ATP production. So this can fuel your neurons during those 3 days that you're trying to track down your prey, okay? Which happens in the real world. But also there's emerging evidence that these ketones have signaling functions. For example, Christina Morosi, who's a postdoc, has found that ketones upregulate BDNF expression. And a paper we're revising, we show how that works. Okay, I'm running a little long, but if I can have 5 more minutes. The last part of my talk is more hypothesis than really convincing data, but hopefully some of this will make sense to you. Okay, plants evolved before we did. I think most of us would agree with that. Okay, so we evolved in an environment where it was to our advantage to be able to consume plants and things like fruits, right? The things we commonly get at the grocery store. But in the wild, that's more like this environment. So here's our ancestor in the canopy. And there's some evidence, in fact, there's a whole book, I think, published on the notion that the prefrontal cortex, part of the driver for its evolution had to do with decision making in our arboreal ancestors on where to go at particular times of year to find particular types of fruits where that particular fruit tree, the fruit was ripe at that time of year. You can, if you want, you can read about that. Just put in prefrontal cortex, whatever, I don't know. Decision making, food selection or something. Okay. Now these, on the other hand, the plants have a vested interest in not being completely consumed, not only by herbivores, but by, maybe more importantly, by insects and other pests. So plants produce natural pesticides, natural toxins. They can't run away from their predators, and instead they produce toxins, which we consume. That is a fact. Okay, might it be that some of these chemicals in fruits and vegetables that evolved, their evolutionary function as toxins, might those be some of the chemicals that are good for our health? And might they act by inducing adaptive stress response pathways in cells, including neurons? Okay, so I'm running out of time, so I wrote an article for Scientific American on this. It was in the July 2015 issue, so you can just look up and read what I'm going to tell you. So the idea here is that we can consume a certain amount of these chemicals in fruits and vegetables before they become toxic. This is the classic hormesis idea. And it's to our advantage to be able to consume these. So we evolved mechanisms to prevent us from killing ourselves by eating these. One is we vomit. Two is we have cytochrome P450 enzymes in our liver that rapidly degrade these natural toxins. So when we consume a grape or a bitter broccoli, one passed through the liver, a lot of it's metabolized, the potentially toxic substance. Okay. Now, what are the first cells are some of the first cells that encounter these chemicals when we ingest them? In insects, it's sensory receptors called sensilla, so neurons. In us, it's our taste buds. There's pretty good evidence that most of the chemicals that seem to be good for our health have a very bitter taste. Caffeine. Caffeine is a toxin. In fact, there's been lately some kids in the United States who have died from consuming too much of these energy drinks. If you put caffeine on your tongue, pure caffeine, have you ever done that? Anybody? Do it. You won't want to eat it very much. So, and on the other hand, from an evolutionary perspective, it's important that we be able to tolerate these chemicals because these plants have calories and other nutrients that are good for us. So anyway, this capsaicin is in hot peppers and things like epicatechins in dark chocolate. Have anybody eaten 100% chocolate? 85%? Okay, try 100%. It's even, it's very bitter tasting. It's hard to eat. Okay, and then in some cases we know the pathways that are activated. Caffeine's a good example. Calcium release from the endoplasmic reticulum, inhibit phosphodiesterases, increase cyclic AMP, so calcium, cyclic AMP, Krebs. That's why it's good for your neurons. So I think I don't want to run over time, but I found this book called Insect Antifeedants. It has like 700 chemicals that have been isolated from some plant and shown to have a noxious effect on insects. We were able to obtain about 50 of these chemicals, and we had tested their ability to activate known adaptive stress response pathways in neurons. One's called the NRF2-ARE pathway, which is known to be activated by sulforaphane and other broccoli things. NF-kappa B and FOXO. We found one, plumbagon, that's really good at activating this antioxidant response pathway, and then we showed this plumbagon is protective in a stroke model. So this is my last slide. Okay, so I've been talking about some of the pathways that are activated during and probably continuing for some time period after these challenges. Okay. However, a recovery period is important. A lot of literature coming out now on sleep, the importance of sleep for the brain, and the reason is because during the stress, overall protein synthesis is downregulated in cells. mTOR is downregulated, overall protein synthesis is downregulated. The cells go into a kind of a stress resistance mode. So that's during the exercise, during the fasting, during exposure to the natural phytochemical toxins. Then during the recovery period, like, rest after exercise, eating after fasting, sleeping, then mTOR is upregulated, protein synthesis is upregulated, your neurons grow, form more synapses, neurogenesis probably occurring. So we think these cycles of challenge recovery may optimize brain health and perhaps decrease vulnerability to disease. So I'm not going to, I mentioned a few of the people who did the work where I showed you the primary data. The other people are acknowledged in the publication. So thank you very much.